First of all, thanks to the organizers for having me here. I really like seeing what other groups coming from physics are doing in biology, especially what students are doing in different groups. It's always a lot of fun for me to look at. So the topic that my group, as many of you, my background was in theoretical physics, and I got, during my PhD, got drawn to the topic of gene regulation. And the reason I got drawn to this topic is that gene regulation is a key ingredient to many of the behaviors, the complex behaviors that distinguish living matter from non-living matter. So, for example, it gives biological systems a sense of agency. They allow cells to respond on their own behalf to their environment. So in this simple example, there's this bacteria living in a blue environment, and then when the environment changes to orange, the bacterium somehow senses this and then starts expressing some genes that make it color green here that allow it to divide in this orange environment, which it didn't allow it to divide when these genes were not expressed yet. So in this, it really makes sense that the bacterium is turning these genes on so that it may divide in this environment, right? It makes sense to talk about there is a goal that the bacterium has, and this is something specific to biological organisms. Second, it allows multicellular organisms to express very diverse phenotypes from a single underlying genotype and to create these sort of societies of cells where they all work together. At the same time, the physical mechanisms underlying gene regulation are rather simple and in principle relatively well understood. So basically, there are DNA binding proteins and RNA binding proteins that recognize particular sequences in the DNA or RNA, and the constellations of these regulatory sites determine the binding patterns, and these determine gene expression levels. So it also means that the sort of gene regulatory network is encoded in these constellations of regulatory sites on the DNA. And so when mutations come and change the DNA, they're sort of directly acting on gene regulation. And so this means that you have some chance to maybe study how gene regulation evolves. All right, so this is what attracted me to this topic. And what I'll talk about today, I'll hope to talk about two topics. The first is on how gene expression noise helps gene regulation evolve. So this is published work, which is a few years ago, and I'll spend the first half on this. And the second half, I want to talk about some unpublished work on using single-cell RNA-seq data to infer something about epigenetic landscapes. All right. To give some context to the first topic, so already a long time ago, I discovered that the number of genes in bacterial genomes that are assigned to different functional tasks scale as power laws in the total size in the genome. So in this plot, this is log axis. On the x-axis is the total number of genes in the bacterial genome. On the y-axis is the number of genes in different functional categories. So in blue is, for example, the number of metabolic enzymes in different genomes. In green, the number of genes involved in translation. And in red, the number of genes involved in transcription factors. So what you should see is that, well, you get straight lines. So that means this number of genes in different functional categories grow as power laws in genome size. But the thing that I want to sort of focus on here is that the steepest slopes that you find are for regulatory functions, for transcription regulation and signaling genes. That is to say, as bacterial genomes get bigger, every time the genome size doubles, the number of regulatory proteins you have quadruples. All right, so expansion of genome sizes is to a certain extent driven by expansion of regulatory capacity. All right, so this raises the question, when does natural selection actually select for increased regulatory capacity? Under what conditions will bacteria evolve to get more complex regulation? And how does regulation evolve de novo? So if you start thinking about it, this is actually a question that there's not been a lot of work on. So when people study gene regulatory networks, it's typically implicitly assumed that the interactions between regulators and their target genes have been finely tuned so as to give the appropriate regulation of gene expression. But the question is, how would you get there if you start from a place where there is no regulation at all? Right? So imagine that there is some signal in the environment that previously the bacterium was not aware of at all and was not responding to. And you want to evolve some gene regulatory circuitry to let it respond to that. So a number of things would have to happen. So there would be a regulatory protein whose activity must become dependent on the presence of this signal, either by binding to some small molecule or by some signaling cascade. Then the appropriate target genes in the genome would have to evolve binding sites for this regulatory protein, avoiding to evolve binding sites in target genes that don't want to be regulated. And then the strength of these interactions have to be tuned so that these different targets are up or down regulated to the right level. So that seems to be a lot of things that have to happen before it's really beneficial to you. So you may ask, how does this come about? Yes?
Can you imagine an organism that has a lot of proteins for translation, metabolism, without any regulation? Because I have the impression you are putting two different categories, like in this plot.
Yeah.
Like in the translation factor,
yeah.
will be the one participating to the regulation, but the metabolic enzymes or the translation, or the translation proteins, they could also be regulatory proteins.
No. Well, okay, so these are the proteins that have a DNA binding domain and bind to the DNA. These proteins don't have a DNA. But they have a known enzymatic domain. So, does that answer your question? Yes. And these lines you find for almost any high-level functional category. So what I'm showing here on the right are exponents of these power laws for different functional categories. Well, okay, so here you get really down to it just 2 or 3, right? And at that point it also starts becoming a matter of definitions, right? So in biology, so do you call the RNA polymerase a transcription factor? Okay, so then you get into questions like this. But you really get down to a very, very small number, whereas in the big, this is not up to date. In the biggest bacterium, there are like 1,100 transcription factors, which is like 50% more than fly, which have like 800. So it's really going over a wide range. All right, so the topic is how does gene regulation evolve de novo? And I'm going to try to show you some insights that we gained from a study that was really a study on gene expression noise. And I'll tell you what the answer is going to be. The answer is that gene regulatory noise, gene expression noise, helps gene regulation to evolve de novo. All right, so before we delve into this, I want to just sort of set the stage by giving you a couple of numbers for E. coli, which is the organism we're going to look at. And so first here is across different genes, what is the distribution of the average number of mRNAs per cell? So this is on a log2 scale. So 0 here means 1. This is half, a quarter, an eighth, and so on. So you see that for the large majority of genes, the number of mRNAs that are on average in a cell is less than 1. So most of the time there's no mRNA, then there's 1 for a while, then there's no. Okay? That's the regime bacteria are in. mRNA half-lives, they have some distribution, but it's very short, just a few minutes. And the mean protein number for all genes, you see here, it goes somewhat from 1 to 1,000. For essential genes, it goes from maybe 10 to 1,000. They tend to be higher expressed. All right. So, and then also note that the DNA is typically, of course, only present in 1 or 2 copies per cell. So it's clear that we're in the regime of very low molecule numbers, and so you expect gene expression to be noisy and stochastic. So if you think about the absolute simplest model, the absolute simplest model would be there is a constant probability per unit time at some gene that the RNA polymerase will come and make a new mRNA. And for each mRNA, there is a constant probability per unit time that it decays. All right? So if you do that, then the steady state distribution of the number of mRNAs per cell will be a Poisson distribution, where the mean is given by this ratio of the transcription and decay rate, and the variance is also given by the ratio of the transcription and decay rate. Now, if you add translation, so you assume that each mRNA is also translated at some constant rate, lambda p, and each protein is decaying at a rate lambda p, you can then work out what is the limit distribution of the number of proteins per cell. And okay, so to do this analytically, you have to make some small assumptions. So for example, an assumption is made that proteins are long-lived as relative to mRNA decay, which is almost always true. And then if you do that, you get a negative binomial distribution, which is controlled by two parameters. A is the ratio of the transcription rate and the protein decay rate. So that is how many times do you transcribe in the typical lifetime of a protein. And second, what's sometimes called the burst size, how many times is an mRNA translated before it decays? Okay? This is an error. This should be mu r. I'm sorry. All right, so these two numbers determine this distribution. And if you now look at the mean and variance, so the mean is just the product of A and B, and the variance is equal to the mean, but now not multiplied by 1, but by B plus 1. So basically, this translation of mRNAs into proteins is sort of multiplying the Poisson noise in the mRNA number by a factor B, which is how many times a typical mRNA is translated before it decays. So important for the rest of the talk is that that means that the coefficient of variation squared, so the variance divided by the mean squared, is this B over n. So it drops as 1 over the average expression. Okay, so because of this noise proportional to the mean, because of this variance proportional to the mean, this noise level drops like 1 over mean. So about 15 years ago, some people started systematically measuring gene expression noise in single cells. So this is a very famous paper from Michael Elowitz, where he put on the E. coli chromosome 2 fluorescent proteins, one red and one green, that were controlled by the same promoter, and then made pictures to quantify the amount of red and green in each cell. All right, so one of the things they did, for example, was then to define these notions of intrinsic and extrinsic noise. So you can look at the total variation in the amount of red and green fluorescence across cells, and you can write that as a sum of two terms. One is the average square deviation between the amount of green and red in each cell. All right, so in the same cell, of course, these two promoters experience the same environment. So you can ask how much variability is there in expression in the same cell, and the rest, which is basically the total minus this, which you can work out in the limits of a large number of cells, is essentially just the correlation of the amount of green and red across cells. Now, in the very simple model that I just told you, where each gene is just transcribed at a constant rate and translated at a constant rate, there would be no correlation, right? There would be no correlation in the amount of green and red in a cell, and so the extrinsic noise would be 0. But if you look at the data, that's not what you find, right? So this is green and red, and so you see that there is this correlation, there is this extrinsic noise, and in fact, in many conditions, this extrinsic noise may in fact dominate the total amount of noise. So here you see, for example, as you induce one of these promoters, its intrinsic noise is really dropping like 1 over n, as I showed you, but this extrinsic noise is showing a maximum and then going down. So it's not so hard to understand why you see this behavior, because we know that promoters are not transcribing at a fixed rate, but the state of the promoter can change, right? So it can be unbound, the RNA polymerase can bound, different regulatory proteins may bind and unbind from the promoter, and in each of these states that the promoter can be, you will have a different rate of transcription, right? This is a simple sort of idea of what's going on here. And as these promoters are switching their states, they switch their rate of transcription. And of course, these switching rates depend on the concentrations of DNA binding proteins in the cell. And so the states of the promoters of the green and the red one will be correlated across cells, and that will give you this correlated extrinsic noise. All right, so with this introduction, it's now maybe interesting to say, well, let's just look systematically at all promoters in E. coli and measure how noisy each of these promoters is. So that's one of the things we did. So there was a library made by the group of Uri Alon, where they took every E. coli promoter. So you take every region between two operons, cut it out, put it on a plasmid in front of a fluorescent protein. And so you can use this library to basically measure gene expression noise in all E. coli promoters. So by simply measuring the amount of fluorescent light across cells. And you can, so of course you can take pictures on the microscope, but to do this in more high throughput, you can use flow cytometry. So you use a Fox machine. So these bacteria, they in single file fly in front of a detector that excites the fluorescent proteins, and then you measure the amount of fluorescence per cell. And for what I'm going to say next, it's important to know that these machines, they are sorters. So depending on the amount of fluorescence it measures for a cell, it can electrically charge these cells and sort them into different buckets. So you can select cells that have expression in a certain range. Yes?
The regime you talked about was very low numbers, and now they have it on plasmid that is quite a lot.
So this is the lowest copy number plasmid that exists, or that is known in E. coli. So we now know from a lot of measurements and comparing chromosomal and that there is 5 copies per cell of this plasmid. And the copy number, it looks to us actually that the copy number of the plasmid as a function of cell size is more tightly regulated than the DNA chromosome copy number per cell, right? Because as the cell grows, you also go from 1 to 2 copies of the gene. And so as the cell grows, you also go from 5 to 10 copies of the plasmid, and then you divide them in half. And so, so we know very precisely now the difference between, so I can guarantee you that there's no cheating with plasmids here. So to answer Alexandra, okay, the plasmid is not changing the, all right. So if you do this, so here are 2 examples, 2 promoters where we made pictures under the microscope, and we also show you the distribution of log fluorescence according to the Fox machine. And we basically find that for almost all genes in E. coli, these distributions look Gaussian on a log scale. Actually, when we started, we wanted to look for bimodal or multimodal, and it's almost impossible to find them. Okay, so flagella and maybe 1 or 2 other things. But everything else, you get relatively simple log normal distributions, Gaussians in log scale. And so you can characterize each distribution by its mean and variance. And so next, I'm going to show you a scatter where every dot is an E. coli promoter, and along the x-axis is the mean log fluorescence, and along the y-axis is the variance. So for the rest of the talk, so here this line indicates what is the fluorescence of a cell that doesn't have any. Yes?
Is this the concentration of the molecule or the total number?
Total. Total number.
Okay, so I mean, the cell's growing.
Yes.
All the machinery is responding to the fact that the cell is growing.
Actually, in bacteria, to a first approximation, cells are expanding exponentially, and the concentrations of everything stay on average the same. Everything is going up exponentially at the same rate. Now, of course, it can't, because at some point the DNA has to go from 1 to 2. Okay, so there's at some level this has to break down. But it seems that the noise on mRNA production and so on is okay. So Okay, so did it answer your question or not yet? Okay.
Well, what you said raises another question, which is it easy to understand why everything goes together? But maybe, just to be sure I know what I'm looking at, if it's the total amount, then this includes variation. The variance includes variation in cell size.
Yes. So we know that depending on whether you have a high expressed or a low expressed gene, we can now do this in microfluidic devices and track everything in time and so on. I'm not going to talk about this, but so I know that if you look at concentration, the variance in concentration is a lot less than the total variance in fluorescence. But they are related, right? So if a protein is coming out highly noisy here, it's going to also come out highly noisy in concentration because these cell size fluctuations are the same for everybody. All right, so this is the fluorescence of an empty cell. So this autofluorescence, this is twice autofluorescence. In this range, the fax machine is doing something to the signal that we do not understand, and we have not been able to convince the company to find us the person that can explain us what the fax machine is doing to the data. They're just not used to people caring about quantities so much. But above this, when the fluorescence from the GFP is at least as big as the autofluorescence, we can look at this data, and we see that, first of all, there is the variance in log expression tends to go down with mean. But different promoters of the same mean can have very different noise levels. And finally, there seems to be this very sharp lower bound here. Now, this lower bound can be fairly easily understood by these sort of arguments I showed you before, right? So I showed you if you have constant rates of transcription, decay, and translation, you'll get this mean and variance. But if you now assume that these rates are additionally fluctuating from cell to cell, you get another term to the variance, which is basically how much these two vectors are fluctuating times the mean squared. All right? If you then also add the background fluorescence that is approximately constant, and you work out what is the expected variance in log expression, you get this expression here, and that's what the red curve shows. So there's basically two unknown quantities that are fitted here. This is a minimal value of this variance, and it's this curvature B. All right, so we think we understand well what is the lower bound in gene expression noise that any promoter will have as a function of its mean. So you can remove this, and so you now have an excess noise level that is sort of how much more noisy is a promoter than the lower bound of this mean. And so basically what I just want you to focus on is that most genes are sort of bunching up here at this low excess noise, but there is a whole bunch of genes that have much higher noise levels. So the question is, why are these, how has selection acted on these noise levels? Is this just drift, right? Is it that some genes are just more noisy and selection doesn't care about it? Is there maybe that they have been selected to be high noise? And for all these guys, at the minimum, is this because selection has tried to minimize gene expression noise, or is something else going on? So to answer this question, you would really like to know, well, what would be the noise of an E. coli promoter if I had not selected it on its noise properties? So to do this, what we decided to do is we decided to evolve synthetic promoters in E. coli de novo, starting from completely random DNA. So what we did is we took these same plasmid with GFP, but instead of putting native E. coli promoters upstream, we made a library where we put just completely random DNA of about 100 to 150 base pairs long upstream. We made a population of these cells, and then we set a target expression level, and then we went in the fax machine and used this cell sorting ability to basically select from a population of cells the 5% in each generation that are closest to our target level. Then we take the plasmids back out, we take these inserts out of the plasmid, then we do error-prone PCR on these just 100, 150 base pair pieces, we put them onto fresh plasmids, we put them into fresh cells. So we make sure that the only thing that can evolve is that 150 mer, nothing else can evolve, and then we throw it to a second round of the fax, and so on. So here in the top, you see the distribution of fluorescence of the cells in the population from the first to the second to the fifth generation when we're selecting for an expression level that is here. And so this first generation is mostly background fluorescence, but there is a little bit of expression in the tail. But you see already in the second generation, it's shifted over. And by the third generation, you're essentially arrived at the final distribution of expression levels. If you ask for a very high expression level, it takes you 5 generations to get there. But once here in this final population, we have a population of promoters, many of which express at higher levels than any known E. coli gene. Okay, so it's really easy to start from completely random DNA and evolve in just a few generations promoters that express at your desired level. Yes?
What happens if you continue the generation but without selection?
Well, I mean, if you keep doing error-prone PCR, you start breaking your promoter, so that you will eventually shift back down to this initial distribution. It's not something we tried, but I'm fairly confident that's what would happen.
What's the ratio of the error rate in PCR versus the natural error rate?
Oh, so where we do this, there is about 1 mutation per segment on average. So it's really high, because in E. coli, the mutation rate is 10 to the minus 10. So in a 100 base pair, it would be 10 to the minus 8 per generation. So we've multiplied by 10 to the 8, essentially. I can also tell you that actually it turned out this mutation is not important. Almost all the selection is acting on the variation that was present in the initial random library. It's sort of like 1 in 500 of the random DNAs acts as a promoter. This is a bit harder to find. But all the error-prone PCR and stuff, we could have done without it and gotten similar results, turns out.
But there is no real evolution in that sense. So they don't have any advantage in making this GFP, quite the opposite, right? So it's a burden for them to make.
Yeah, but it's very small. We can't, I mean, it's a very small burden. But here's the selection, right? They have to make it through the FACS.
Yeah, so the selection is manipulated.
Yeah, we select. Yes, we select. Yes, there's no natural evolution. Right. All right, so now what we wanted to look at is what are these noise levels of these synthetic promoters. So we took clones from this population of evolved promoters, and then we put them through the FACS to look at what is their mean invariance. And that's shown here as these red dots. All right, so to our surprise, we had expected that because these promoters didn't have a lot of time to optimize their noise levels, that they would be very noisy. But in contrast, what we find is that they're almost all low noise. If you look at the cumulative distribution of excess noise levels, you see that it's in fact native promoters that are enriched for high noise levels. So now what I will skip is you can do an analysis and say, well, didn't you sort of implicitly select for no low noise level in the FACS by setting these very narrow gates and so on? So we spend a lot of time studying that and doing extra rounds of selection and see what happens and so on. So you'll have to take my word for it that in the settings where we're doing that, we're not effectively selecting these promoters on their noise levels. They're much more selected on their mean, and the effect of the selection on the differences in the noise are negligible. What is happening is that low noise is the default behavior of E. coli promoters. If you ask an E. coli promoter to just express at one level, then the promoters that do that, starting from random sequences, are by default low noise. In fact, I now know it's actually quite hard to make a high noise promoter. Yes? Yeah, very little. So all these, even the lowest noise possible promoters, there's a low bound. They are so wide relative to this sort of selection that the relative differences of making it a bit wider or less is very little. Just moving your mean around a little bit is a much bigger effect on the fitness of a promoter. I'll show you in a second. You select on the mean, yes. All right.
If the distribution is very broad, then moving the mean also makes much difference.
Yeah, but it makes more difference than... So what we find from this, the fact that the native promoters, or a subset of native promoters, is more noisy than the synthetic promoters must mean that in the wild, selection has somehow acted on these promoters so as to increase their noise levels. Right? There must have been some sort of selective force that caused the noise of these native promoters to go up. So the next thing that you, of course, then do is you ask what's special about these native promoters that show high noise. And so one of these things is in biology, almost any variable shows some weak correlation with almost any other variable. It's very rare to find things that are completely uncorrelated. So the question is sort of what is really the origin of the correlation? We believe that the origin of the correlation is really this. Noisy promoters have more regulatory inputs. So for example, in E. coli, there are databases with known interactions between regulators and target promoters. And if you sort your promoters by their excess noise and you ask, as I increase excess noise level, what is the mean and standard deviation of the number of regulatory inputs that these promoters have, you see that it systematically goes up. Noise level, you can also look at it in another way. You can split all the native promoters in the ones in black that have no known regulatory inputs, in blue 1, and in red 2 or more regulatory inputs, and look at the cumulative distribution of noise levels. And you see that for promoters that have no known inputs, it's very close to what the synthetic promoters show. And as soon as you have regulatory inputs, the distribution of noise level shifts.
But so all of these regulatory inputs have to work together, or?
No, it only works at the level of the distribution, right? So it's sort of like
I'm just asking really whether the noise is measured, because you couldn't imagine having a promoter which is regulated by different factors in different cases.
Yes.
And then in each case there's some noise distribution it produces. So what is the noise distribution when you take into account all the promoters, or an average over all the conditions?
This is just in one condition.
Okay. Well,
we have now done it in other conditions, but
no, I just mean by condition, I mean, say you have a promoter that has 3
yes.
inputs.
Yes.
But say that one input does functions, they all function alone in, well, I guess, okay, that doesn't make sense.
Oh, it makes sense. In different stresses, for instance, let's say it's regulated under osmotic stress with this regulator in another condition with this other. So I think the point is very good. So that they might be, there are more regulatory elements, but they don't act all in the same condition.
Yeah, I'm just really asking whether how you measure the noise, whether the fact that you have a promoter with, say, 2 inputs in 2 stress conditions, then you can imagine one of them binds, gives you some noise distribution, another one binds separately, gives you some other noise distribution. Is this noise distribution is this excess noise factor an average of those 2 distributions? Or is it?
No, but we don't have such detailed information, right? Okay, all we've done is measured how...
You plot a number, so what's that number?
On the y-axis, the number of regulatory inputs according to the database that this promoter has.
And the x-axis, the excess noise?
How noisy did we measure this promoter to be?
Oh, so you measure it yourself?
I just showed you, right? For all...
You measured all, I didn't understand, you measured all of them in the...
All E. coli promoters. Right, I showed you this big scatter with these many blue dots, that was all E. coli promoters.
So if they have 2 inputs, then you measure them under the 2 inputs?
No. I just measured them in minimal media with glucose in one condition. And then I sort all my promoters by how noisy they are in that condition. And then I asked, these guys that have excess noise of at least 0.2, what is the number of inputs that they have? And I look at the distribution at the number of inputs that they have, and they say, it's like this. All right. So the next question, and now I'm going to start to do a little bit of theory, is so why is there this general association between being noisy and being regulated? And why did selection cause noise to increase? All right, so here's a very simple model of what's happening in the fox. So I have some promoter who's characterized by some mean and some standard deviation in log expression. So the single cells of this promoter have some distribution of expression levels. And now I come with my fax machine and I select promoters that are near some desired expression level mu star. And maybe there is some sort of the broad of the width of the distribution of how much you're allowed to vary in expression is about tau. So the probability for a single cell that expresses at level x to make it through this gate is given roughly by a Gaussian distribution. And so now you can ask, what's the fraction of all cells with a certain mean mu and standard deviation sigma that will make it through the gate? That's just the convolution of these two Gaussians. And so you can easily analytically get this expression. And so you see that basically the number goes down like a Gaussian with the difference between the mean of this promoter and the desired level relative, divided by the sum of the variances of the two Gaussians. And then there is some prefactor. So now I want to ask what would happen if instead of exposing these cells to one environment, I were to expose them to many different environments. And in each environment, E, there is some optimal expression level mu E. What will be now the long-term fitness of a given promoter? And so the long-term fitness is basically the geometric mean of the fitnesses across all these environments, or the long-term log fitness is the average of the log fitness across each environment. So that now this is easy to calculate. The log fitness is basically the mean square deviation between the expression level of the promoter and the desired expression level in each environment averaged over the environments relative to, again, the sum of these variances. So if you have a promoter that's not regulated and you have these environments that ask you to express at different levels, you can ask what's the best compromise that you may find. And the best compromise is, well, first of all, you want to set the mean expression level to the mean of the desired levels across environments. And then once you plug that in, you will see that this is your fitness. So it's basically controlled by the variance in these desired levels across environments. And if you now ask what noise level of the promoter will optimize your long-term fitness, basically the variance of the promoter is equal to the variance of the desired expression levels across environments. So to just give you a very simple cartoon, if this was the expression distribution of the unregulated promoter, and we have these three environments where three different levels are desired, then the best solution for an unregulated promoter is to put the center into the average of these three and to basically make the distribution wide enough such that in each environment there is at least some subset of cells in the population that express at the right level. So now this is what is generally known as a bed hedging scenario. Okay, so you make yourself so noisy that there's always a subset of individuals that express at the right level. But so now the question is, wouldn't it be better to evolve regulation? So let's look at that case again in a simple cartoon. We have an unregulated promoter. There are here three environments that ask it to express at different levels. So this unregulated promoter is not doing so well because it has a good overlap here, but not in these two environments. Now, ideally speaking, there would be some transcription factor in the genome who is upregulated in the green environment and who is downregulated in the red environment. If there is such a transcription factor in the genome, if you now evolve a binding site for this transcription factor in the promoter of this gene, then basically the transcription factor will push it up in the green environment and push it down in the red environment. And now you will have a much higher fitness and even much better than you could have obtained by this bed hedging scenario. All right, so this is the way we think about how gene regulation works. But I want to point out that there are sort of two effects of coupling this promoter to this transcription factor. So the first, which I call the condition response, is that the mean level of the promoter in each condition becomes a function, or becomes correlated with the mean level of the transcription factor in each condition. And this is, of course, the way we think about how regulation works. But on top, because the transcription factor has itself some variability in each environment, this noise in the transcription factor's activity will be propagated to the target gene. All right, so per definition, the noise level of the target promoter will go up when it couples to the noise of the regulator. So this is noise propagation. And so what we did is we worked out how do these two effects, the condition response and the noise propagation, combine to affect the fitness of the promoter, right? So we take the same sort of system that we looked at here, but we now included these regulatory scenarios. And so I won't go through the derivation, but under fairly weak assumptions that you can approximate integrals with Laplace approximations and so on, you can actually analytically derive an expression for the effect of coupling a promoter to a regulator. And it turns out that this effect on long-term fitness is a function of only 4 effective variables. So the first effective variable we call expression mismatch, that basically measured how much the desired expression levels vary across environments relative to the variance of the unregulated promoter. It sort of matches how unhappy an unregulated promoter is, how many standard deviation it's away from the desired level on average. The second is the signal to noise of the regulator. How much does the level of the regulator vary across environments relative to how variable is the activity of a regulator in each environment? Third, and this is kind of obvious, you'd like to have a regulator who goes up and down across environments in the desired way. So you can look at the simple Pearson correlation between the expression levels of the regulator across environments and the desired levels across environments, and then finally the coupling strength. So in terms of these 4 variables, you can write down an analytic expression for what's the effect of evolving a binding site for a regulator. So to show you what this theory predicts, I will start with an unregulated promoter and then look at what fitness can be obtained by coupling to a regulator that has a certain signal to noise and a certain correlation R, and so I'm going to optimize this coupling strength. All right, so along this axis is the signal to noise of the regulator. So these are very noisy regulators. These are very precise regulators. Along this axis is the correlation. So here at the top, the regulator does exactly what is desired. It's going up by and down across environments by the exact amounts that are optimal. And at the bottom here, there is no correlation between the regulators and the desired level. All right. So, and then the surface shows the fitness you can attain, and for reference, this is the fitness of the unregulated promoter. So not surprisingly, the best fitness you can attain when there is a regulator that has a very high correlation with the desired levels and that is relatively precise. So here's a cartoon example of what this would do. And in this regime, the increase in fitness is completely a result of this condition response, right? So the regulator is pushing the promoter up and down in a condition-dependent way, in the way you want. And there is a little bit of increase in the noise level of the target promoter because of noise propagation. But here, noise propagation is just an unavoidable side effect of the coupling of the regulator to the promoter. However, if you go all the way here, we have a promoter that is, we have a regulator that is very noisy and that doesn't correlate at all with the desired levels across environments. When you couple to this transcription factor, essentially what happens is that you just make your promoter very, very noisy. So by coupling your promoter to this regulator, you use the noise propagation to become very noisy, but that effectively acts as a bad hedging scenario in this case. And so it's also beneficial. All right, so you can already increase your fitness a lot by simply finding a regulator that's noisy and coupling to it and making you more noisy. No, because, so basically what we found is that if you're in E. coli and you want to be noisy, the way you do it is you find a regulator that's noisy. There is no, you need to make your promoter change states, because otherwise it will just express at a constant rate. And to make your promoter variable, the easiest way to do it, that's what the experiment has shown, is to evolve binding sites for noisy regulators.
I thought that you showed, but maybe I'm wrong, so that it was without any regulation, the strategy would be I select some sort of...
Well, that would be the optimal strategy, yes.
And this was the bad hedging, without regulation, so the same.
Yes, so this is the bad hedging, but you use the regulator, you use noise propagation to implement the bad hedging. There is no other simple knob E. coli can turn to become noisy. It uses regulation to become noisy. That's in fact, so that's one of the key points. Huh?
To say it more precisely, what you're saying is that it uses its extremely reliable response to a regulator whose concentration is fluctuating.
Yes.
So,
or I mean, actually, we don't. This is something we're studying right now. Or is basically the regulator is at a level where the binding to the promoter fluctuates a lot from cell to cell. So we don't really know if it's concentration fluctuations that dominate, or if it's state fluctuations that dominate.
Dumb question. Why
Yeah.
is better doesn't do on your color map?
Huh?
Why is better thickness than blue?
Then?
No, like the color map,
Yeah.
the color code on your thickness map. Blue is lower thickness
Yeah.
than white.
Yeah. So this is this is lowest and this is highest.
Yeah.
I'm sorry. Was this unclear? Blue is deep and so yeah.
All of those E. coli make noisy transcription factors in the first place.
Yeah. Okay. By coupling to a noisy transcription factor, all the way through. No. I mean, there are signals in the environment, right, that will fluctuate.
So it's coming from outside of the grid. It's using information from the outside.
In the end, yes. But as I also, in my response to Bill, we don't really know if it's really the concentrations of the transcription factors that fluctuate, or there might be some transcription factors who are at a level that's very similar in all cells, but at that level, the promoters fluctuate a lot between being bound and being unbound.
So why would you want more than one?
Well, I mean, there are still different signals that you respond to, right? I mean, still the best is to have good regulation.
Okay.
That's still the best, of course. It's just saying you can already get a lot of bang for your buck by just making yourself more noisy by finding a transcription factor that's noisy. All right, so I think what sort of surprised us in the beginning is that there are even cases where you take a regulator that is moderately correlated with your desires, but the fitness that you can attain by coupling to this regulator is actually lower than the fitness that you can attain by coupling to a regulator that is totally uncorrelated with your desires. And the reason is that this regulator is too precise. All right, so the distribution of activity of this regulator in each environment is rather narrow, so it pushes you around, but it pushes you around too precise. And therefore, in some conditions where it's not doing what you want, most of your distribution is not overlapping. If you were to lower the signal to noise of this regulator, if you made it more precise, then you would actually do better than with this regulator. So now you can ask, at each correlation level, what's the optimal noise level of the regulator? So that's what this plot shows. So basically, at each correlation, you ask, what would be the signal to noise ratio of the regulator that gives me most bang for my buck? And that gives you this line. So on the right of this line, regulators are not noisy enough. They're too precise. On the left of this line, regulators are too noisy, and the noise propagation acts to make you less accurate than you want. But along this line, the noise propagation and the condition response are sort of optimally working in concert. And this basically shows you now scenarios how you can go from no regulation at all to precise regulation, right? So you can start here on the bottom left by first coupling to a regulator who is mainly just acting to make you more noisy. and implementing a bad hedging scenario. But now there is a whole continuum where you can start tweaking the expression levels of the regulator in each environment so as to increase its correlation and slowly decreasing its noise level. And in this continuous way, you can make it up all the way here, where now you have a very precise regulation. All right, so the summary of this is that noise propagation is often functional, and you can think of it as a rudimentary form of regulation, just shaking you around a bit. And you can start from pure noise propagation and in a continuum of solutions increase accuracy along which condition response and noise propagation complement each other. And the reason you expect E. coli to be in a regime where you use noisy regulatory strategies is because you can attain fitnesses that are very close to the optimal fitness you can attain with a perfect regulator by having a regulator that is just roughly pushing you in the right direction and adding some noise to it, and the fitness you can attain with such a strategy is already very, very close to the best fitness you could possibly attain.